Alright, this is going to be the last portion of the lecture for chapter 22. We are going to talk about glycogen metabolism, and specifically glycogenesis and glycogenolysis. So before I fill in any blanks, I want to draw your attention to the figure this time. This is a different figure than the one up earlier in the lecture slides. Okay, this is its own unique figure. It looks a whole lot like that other figure with glucose and glycolysis, but it's its own figure specifically for the formation or the breakdown of glycogen. So you do not have to memorize this one or spend time studying this one the way I encourage you to with that figure above. I think it is a really helpful reference to kind of know where you are in the pathway though. But I just wanted to point out that difference. It's not the same figure, it's a new figure, and we're going to look at it in a second. So glycogenesis is the biochemical pathway for the synthesis or the formation of glycogen. And what is glycogen again? It is a branched polymer of glucose. It is how the body stores glucose. So there's some important enzymes in this pathway, and any name you see that's italicized is going to be an enzyme here. But I want to start up here with glucose. We'll get to those enzymes in a minute. So remember, glucose is first, when it crosses into the cell, okay, so when it crosses into the cell, we immediately phosphorylate it. Hexokinase is the enzyme that does that. We're actually not committed to glycolysis at this point in the pathway. Step 1 does not commit glucose to glycolysis. You're not committed to glycolysis until you isomerize into fructose 6-phosphate. All right? Once you isomerize to fructose 6-phosphate, you are now committed to glycolysis. That's the route you're going. But glucose 6-phosphate could have another fate where this enzyme right here, phosphoglucomutase, converts it to glucose 1-phosphate. So all we're doing is moving the phosphate group from carbon 6 to carbon 1. That's what those numbers indicate. All right, so that's what you read over here. Phosphoglucomutase isomerizes G6P, glucose 6-phosphate, to G1P, glucose 1-phosphate. So we're going to move that phosphate group around. So it's actually this phosphate group right here. It was on this carbon, and now we've moved it to carbon 1. Right? And the reason we're going to do that is because we're getting ready to attach it to uridine triphosphate, very similar to ATP, adenosine triphosphate, but it's uridine, it's a different base. And we're going to UTP, again, this is a triphosphate, and we're going to take off 2 of those phosphate groups, as you see here, and then we're going to link up glucose to it, and so we call it glucose UGP, which is actually this whole molecule right here. Okay, so here's glucose, right? And then you see the 2 phosphate groups in the middle, which is a diphosphate, so that's why we call it UDP glucose. And this is the activated carrier of glucose in glycogen synthesis. So this is kind of like its chaperone or its chauffeur to get to glycogen. All right, so UDP is going to, we're in the liver now, by the way. This is where, I'm going to write that down here. The liver is where we store glucose as glycogen. And essentially UDP is just going to drop glucose off into its glycogen polymer. And what does that look like? It looks like this. You've seen this figure back in chapter 20, the very end of those slides. It is going to link the glucose monomers together, the monosaccharides, via these alpha-1,4 glycosidic bonds. And you can also branch off carbon 6. And all this branching kind of ultimately gives you a structure that might start to look like this in mammals. Okay, it's this very branched kind of looking structure as it grows and forms and stores more and more glucose. All right, so that's the formation of glycogen. That is glycogenesis. All right, that's the blue arrows, and this is anabolic. So now let's talk about the catabolic part, the breakdown of glycogen back to get readily available forms of glucose. All right, and that is known as glycogenolysis. Remember, lysis is like you're lysing, cutting something down, breaking something down, and synthesis, or genesis, is the word for synthesis. All right, so glycogenolysis is the biochemical pathway for the breakdown of glycogen to free glucose. And there's a couple key players here glycogen phosphorylase, which is listed right here. Glycogen phosphorylase is the main enzyme that is going to come over here and it's going to cleave these alpha bonds so that you get a free monomer. And it's also, at the same time that it cuts the glycosidic bond, it's also going to phosphorylate glucose simultaneously. It's a really, it's a powerhouse of an enzyme, doing two things at once, cutting the glycosidic bond and phosphorylating glucose simultaneously. So you see this phosphate group is also necessary. And then you end up with, when that's all said and done, you remake glucose 1-phosphate. And then glucose 1-phosphate can be converted to glucose 6-phosphate from there. And let's see how that happens. Oh, sorry, actually a couple more things about glycogen phosphorylase. It is formed as a zymogen in its asleep inactive form, and then it's activated via covalent modification. By a kinase enzyme. What does a kinase enzyme do? It phosphorylates something. So glycogen phosphorylase itself has to be phosphorylated to be turned on, or waked up, to be awake. And so there are actually two forms of phosphorylase. The active form, also known as phosphorylase A, the inactive form known as phosphorylase B. And then this last thing down here, there's two distinct forms. Now, I'm not talking about A and B here, the active and the inactive form. I mean, there's two actual unique forms depending on what tissue you're in. So there's one type of glycogen phosphorylase in your muscle tissue. There's another kind in your liver tissue. We're going to talk about why that is. Why do we have two forms of this? And then lastly, the next part of glycogen metabolism, once glycogen phosphorylase has done all its work, this guy, phosphoglucomutase, isomerizes G1P to G6P, and then it can be ready to enter glycolysis if it wants, or if you are in the liver, you won't go to perform glycolysis, you will just then be dumped right back into the blood. All right, so this is where these two enzymes we just talked about, phosphoglucomutase and glycogen phosphorylase, these are the two major players in glycogenolysis. All right, let's talk a little bit more about the difference between glycogen phosphorylase in your skeletal muscle and in your liver. And so I'm going to talk about the kind of allosteric modulation of this enzyme. You see all these little activators and inhibitors of glycogen phosphorylase. Why is it different in the muscle versus in the liver? And so I want to talk about the muscle first. Okay, so I'm going to be down here. During a sudden intense like exercise, when your muscle is contracting, think about what you're doing. You're using a lot of ATP. So in a resting muscle, we have good supplies of ATP, and we don't need to make any more ATP. So glycogen phosphorylase is really, really sensitive to your levels of ATP. And when we have good supplies of ATP in a resting muscle, so I'm going to write that here. ATP acts as an inhibitor to glycogen phosphorylase, okay? Another inhibitor would be glucose 6-phosphate. Why would glucose 6-phosphate inhibit glycogen phosphorylase? Well, let's go back up to our figure. Okay, glucose 6-phosphate is right here. And glucose 6-phosphate can directly feed into glycolysis. So if we have a good amount of glucose 6-phosphate, like in a resting muscle, then we don't need to make more glucose. We don't need to make more ATP because we have a good amount of it. So we don't want to sit here and run all of glycolysis. So glucose 6-phosphate is the perfect inhibitor to glycogen phosphorylase because remember, you're not committed to glycolysis until you become fructose 6-phosphate. And so this is an excellent inhibitor. That way we don't accidentally turn on and run glycolysis if we don't need to. So glucose 6-phosphate and ATP are both allosteric inhibitors of glycogen phosphorylase. Now, I also want to talk about calcium because you see that calcium is an activator of glycogen phosphorylase. Oh, and one more. There's another one. AMP. So remember, AMP and ADP are byproducts of ATP. So when ATP is converted or dephosphorylated, what you end up with is ADP. So when you remove a phosphate group off of ATP, you get ADP, and you can further dephosphorylate ADP into AMP when you lose another phosphate group. And so the muscle is really, really sensitive to levels of AMP. That means we're running out of ATP. Now remember, your muscle is low on oxygen, so we get most of our ATP from anaerobic conditions. Most of our ATP is produced directly from glycolysis under anaerobic. Whoops. There we go. Anaerobic conditions. And so ATP is produced directly from glycolysis. And because of that, this glycogen phosphorylase is going to be really, really sensitive to levels of AMP. So when we are low on ATP, AMP is going to be present in a really high amount, and it's going to turn on glycogen phosphorylase so that we rev up production of ATP via glycolysis in our muscle cells. Okay, let's also talk about calcium. One of the subunits of glycogen phosphorylase is, where should I write this? Is calmodulin. You actually don't need to memorize that, so I'm not going to write it down. But calmodulin is a spot where calcium binds calmodulin, which is a part of glycogen phosphorylase. It's like a subunit. And why would that turn glycogen phosphorylase on? Well, calcium, when your muscle's about to contract, the levels of calcium in your cytosol, there's like an abrupt rise and increase in calcium. And then that's an indicator to the muscle that it's about to do some work. It's about to contract, okay, which is a signal to the body to go ahead and start breaking down glycogen so that we can start creating more ATP, because the muscle's about to do some work, and we are going to be in a deprived state of ATP after that work, and we need to be ready to replenish the amount of ATP once our muscle gets done working. So calcium really is here kind of as a signal and a sign to this whole pathway to support muscle work by creating more ATP, because it's a sign, one, that the muscle is getting ready to do some work, and that's going to require a good amount of ATP on the body. So, up here in the liver. So the liver is a little bit different. Liver cells, hepatic glycogen phosphorylase, it behaves differently than that of muscle. It's not as sensitive to like your levels of AMP. That's because we don't run glycolysis and we're not contracting like a muscle. So really, the major players here, you see all these inhibitors of glycogen phosphorylase. When we have a good amount of ATP, when we have a good amount of glucose in the body, these all come back and allosterically inhibit glycogen phosphorylase, glucose 6-phosphate as well. All right. Now, the difference between muscle and liver really is related to the role of glycogenolysis in each of these tissues. Okay, so in muscle, remember that glycogen breakdown is activated to meet the energy needs of the muscle. Okay, that's down here. But in the liver, glucose is not the main source of energy. And because it's not the main source of energy, then the hepatic or the liver form of glycogenolysis is really just so that we can break down glycogen into free glucose, which we then just dump right into the bloodstream. Okay, so the purpose, let me re-summarize that. The purpose of glycogenolysis in the muscle is so that we can quickly regenerate ATP if our muscle's getting ready to do some work. But the purpose of glycogenolysis in the liver is so that we can make free glucose to adjust our blood sugar concentration, the concentration of glucose in the bloodstream. Okay. If you have questions on any of this, make sure you write those down and send those to me on why this would be different in muscle versus liver. Okay. And then last thing, gluconeogenesis, which is the formation of, the synthesis of glucose from non-carbohydrate sources, right? So this is taking like amino acids or fatty acids, glycerol, and making glucose. So I'm actually not really, I'm going to skim over this. This is not the major player in chapter 22. It's just kind of an end note, side note. Why is it important? So right here, gluconeogenesis is utilized to meet energy demands when stores of glucose and glycogen have all been used. All right. So again, if you're in a state of starvation or fasting or dieting and you don't have any glycogen left, you don't have any glucose left, then the body's just going to pull all its resources and all its reserves from other biological molecules and do everything it can to start producing glucose. Okay. Now, pyruvate is one of the players. You can literally, you see these upward arrows, you can literally run all of this in reverse. A lot of these steps are the same steps and same enzymes and same intermediates from glycolysis. So it's like you're just running glycolysis in reverse to some degree. It's not exactly the same. But you can just run this in reverse and ultimately end up at glucose. Or in addition to pyruvate, you can take molecules like DHAP, okay, dihydroxyacetone phosphate, or glycerol, which, remember, glycerol is the backbone of fatty acids. And these can enter the pathway at step 7. So you can take DHAP, glycerol from fatty acids. I actually put the structure of DHAP right here so you can see how similar it is to glycerol, okay? Because the first step with glycerol is that you convert it into DHAP. Well, how does that happen? Well, look at this. This is pretty much almost glycerol. Let's draw out glycerol and see how they're like almost the exact same thing. Glycerol is just this. You guys drew this when we learned how to hydrolyze triacylglycerols. Okay, so there's glycerol. And look how similar DHAP is. So it's just one, you're just removing these two hydrogens. Essentially, you're oxidizing the molecule. So this is an oxidation step. And you're getting DHAP, which can then be converted right into glyceraldehyde 3-phosphate. We know there's that triose enzyme that does that, and then you can produce glucose this way from non-carbohydrate sources. So this is the major thing I want you to take away from this slide, and I have scribbled all over it. But that gets us to the end of chapter 22, and I hope you send me your questions.